Okay, thanks a lot. I would like you to show you some of the results from the research from the last years working in SmartNanoTox. Our role of our lab, our team, was to enable tracking of the very early events in live cells. So between the exposure and the subsequent events in the adverse outcome pathways. So for us, most of us are physicists, and for us, every motion has its own driving force. So in what we were trying to achieve, we were trying to see the connection between the causal connection between the very early events, and especially, as you would see today, between the membrane rearrangement and the events which followed it. So in this time-lapse movie shown here, this is over a few hours. You can see attack of several macrophages on the epithelial cell. But you can also see many more details. You can see a lot of materials that are moving as a whole, as a large assembly, as a large aggregate, despite that it seems that it's composed from many particles. If I start at very beginning, at the beginning, we started with exposure to epithelium, to a mimic of lung epithelium, which is a living LA4 barrier. And this is high definition and high resolution image done with almost 80 megapixels. 1 pixel is 10 nanometer sized. So we are using high resolution microscopy, fluorescence microscopy. And this allows us to see the details that are normally mislooked by classical confocal microscopy. For example, here on these images, the green color represent the membrane structures, and the red color is nanomaterial. In our case, titanium dioxide nanotubes here. And as you can see here on this small zoom in, you can note the scale. So this is the entire image is scaled on over 2 microns. You can notice here we can see approximately 2 tubes. Tubes are 10 nanometer in diameter and up to 200 nanometers in length. So we can resolve individual nanoparticles in living cells. That's the first message. But the second message is that we can resolve co-localization of both signals, so of nanomaterial signal and membrane signal. And here, this yellow color here means that the membrane is actually, the membrane signal appears more or less within the same voxel as nanomaterial do, which means that nanomaterial is wrapped in membrane. And even more important is that at some point, if you zoom in through these images, you would see, instead of clearly resolved nanomaterial like on the image D, we can see horizontal stripes, and these horizontal stripes are due to the dragging by laser, by scanning laser and STED lasers, which allow us to image beyond diffraction limit. And these actually identify as the mobile fraction of nanoparticles. So it means that we have nanomaterial that steal membranes from the cells, and they can move, meaning that they can actually move the material around the system. Wait a moment. So here. So actually, of course, the first question is how this wrapping is driven. We have heard already a presentation from Nick, and we will heard another one from Sasha. So here, just briefly, the results from Sasha's simulation. You can see in the upper row, you can see binding of phosphates, and in the lower row, binding of amines from lipids onto the TO2 nano surface, so surface of nanomaterial. You can see that both of them, they can make an anchoring point for the membranes, and this is the driving force why this is the driving force for what Nick was explaining before. It can actually drive at least partial wrapping, if not full wrapping. So we have at least local contact between the membrane, between the lipids of the membranes and the metal oxide surface. On the other side, so if the lipids anchor the wrapping membrane, then of course one can quickly imagine that it's not just the lipid that are transferred around on nanomaterial, which is served like a carrier, like a truck. And what we did after seeing these stripes on stat, we transferred the sample from washing the epithelium onto the transmission electron microscope. And now, of course, the TEM, you have a much higher magnification, but it's not in live. You can see a lot of small dots, and these are different proteins. And if we transfer the same, we did transfer the same sample to the proteomics in Dublin. David was doing that, and we identified several hundred different proteins. And interestingly, of course, majority was the plasma from plasma membrane, but there were a lot of other cellular compartments from which these proteins originate from, and that means that somehow, even we wash this material from outside of the cells, somehow at some mean way, this material came out from the cell on the surface of material. And there are some nicely, very highly populated proteins found in this sample, like coagulation factor 10 and tissue factor. So if these proteins, which are now transferred or relocated on the surface of nanomaterial, is relocated over the barrier on the other side, so on blood vessel endothelial, then of course this can trigger at least coagulation cascade, if not something else. And well, how do they can get relocated? Well, you are all aware that the thickness of entire barrier is very small, so it's less than 1 micron. In average, is half a micron, and this is practically the size of few nanotubes. So many nanomaterial have at least one dimension of the thickness of that barrier. So it means that it's really not so far to be traveled for nanomaterial. And beside that, this nanomaterial makes holes in the epithelium, or epithelial cells die, and that's why epithelium becomes porous. There are also like a wormhole structures in this one in the cell. These structures are like tunnels, and they actually allow nanomaterial to pass from one side of epithelium, so alveolar epithelium, to the blood endothelium on the other side. So you can see this, because this is made from actin and nanomaterial, and also membrane is involved. You can see that this is self-assembled structure that create the pores through the cells. This cell, which is here, it's still alive. So it means that such tunnels allow then allow material to pass through. Now the main question is, this phenomena, membrane wrapping, happen at any concentration you delivered. So already at low concentration, it happened fast because it's driven by physical interaction, by affinity between the lipid molecules and the nano surfaces. So obviously, one would think that if you deliver many more materials, this would completely destroy the epithelium. Well, the reality is that the epithelium found a strange way to protect itself. So first of all, if you wait 1 to 2 days, so this is after 1 hour, and this is after 2 days. So this is an image from the top. So you can see many cells, a lot of nanomaterial inside, but you can see a lot of orange, yellow, orange aggregates. And if we made a vertical cross-section microscopy, so this is the lower row, and if we made the dose dependence, then you can see that a lot of material can be seen outside, so on the surface of epithelial cell. And then we can transfer this sample to high resolution methods like scanning electron microscopy or helium ion microscopy, and we zoom in really a lot, and we can see. So here is a typical combination of a surface with those structures and normal bare nanomaterials, and you can see that really here is a huge mixture of nanomaterial and biological material. And because on the left, on this image here, so all of these images here, the green color represent the membrane, and the yellow color means colocalization of membrane and nanomaterial. Like here, if this is a microscopy, light microscopy image, super resolution, you can see that the membrane and nanomaterial builds sort of composite that is now located on the outside. So cell obviously use membrane material to quarantine the nanomaterial which was used in this study. So this is how it survived very large doses locally. And even if you measure cell viability, you will see that cell can manage, the epithelial cells can manage huge doses without even dying. So the question is now, because it seems like a defense mechanism, the question is how can cells survive different materials? So here in this phase diagram, which is on the center of that slide, you can see on the x-axis it's the presence of these composites, agglomerates, however you call them, and on the y-axis you see the survival, the number of viable cells. So you can see that the lowest survival, of course, zero survival is zinc oxide, as we know. There are some structures there. If you coat zinc oxide, then you can see no agglomerates, but also survival is very low. On the other diagonal, it's our titan oxide nanotubes and also quartz silica. And because in this phase space, you can see that this point allows us to study combination, so causal connections between agglomeration and survival of the cell. We actually use these tubes to study further the mechanisms. Interestingly, if you take a look on the in vivo or ex vivo samples, you can see that you can identify similar structures. So this is inverted color from cytoviva images. That's because not to be too similar to fluorescence images. So here the black color is now invert of the pure white. So the black dots is a nanomaterial because it scatters light at all wavelengths everywhere. So you can identify here the blue color here is the epithelial cells, and this violet, light violet or whatever it is this color is, it's sort of organic structure inside the organic matter inside the alveoli. So you can see that we can actually identify similar surface structures all over the sample after the exposure with the same material. As said, not all the nanomaterials harm epithelial cells in the same way, and but it's interestingly that those that have huge agglomerates detected, they have they trigger also strong chronic inflammation in vivo. So that's why it's interestingly locally at the level of epithelial cell, it seems as a defense mechanism, but the level of organism, it still trigger strong chronic inflammation, and we were interested in how that comes out. So we use different, especially different approaches, especially super solution time lapse, to identify the mode of actions. So first, because we co-localize in these yellow aggregates, we co-localize lipids and nanomaterial in the same place. We said, okay, we are interested whether this is a consequence of biological response. So if you might also remember that I said it is 2 days after the exposure. So yes, this because it's so late, we think that the lipids are synthesized as a response of exposure. Indeed, if you block the lipid synthesis, then you see no, at the same time point, you see no quarantine, no these composite structures. And if you do transcriptomics and check the lipid metabolism, you can see that there is huge, the red arrows here, the red arrows all down here, they represent the response of epithelial cell. So they have huge upregulated lipid metabolism, and you can also see the pathways. They're highly upregulated. So interestingly, if you block endocytosis, then similar structures, but much smaller, happen quicker than in half an hour, which means there is a pure physical mechanism that drives the generation of such bio-nano agglomerates, but are very small, much smaller than those several microns large composites. On the other side, if you pull out the cholesterol from the cell membranes, then the nanomaterial is uptaken super fast on the level of minutes, and then it's the nanomaterial cannot be quarantined in the on the surface of the cell, and it's just staying inside the cells. On the other hand, on the right side of this slide, you can see that when nanomaterial get into the cell, it quickly binds to actin fibers. Yes, and indeed, it also it damage or restructure actin fibers. We can see that the actin is actually involved in the exocytosis of the nanomaterial. This is blocked exocytosis after 2 days. So we can identify many of those small vesicles, like here. These are all these exocytotic vesicles with nanomaterial inside and actin fibers around. And you can, even in transcriptomics, you can see that actin elements are also, or production of actin elements are also upregulated. Here is high resolution microscopy of both of the surface. You can see that biomaterial is glued on the top of nanomaterial. And here are the two 3D images from fluorescence, high resolution. The lower one shows you coexistence of actin fibers and nanomaterial. Be careful, this is outside of the cell already, and here is membrane and the nanomaterial at the same outside of the nanomaterial. So actually, nanomaterial, when it is excreted, it actually take over a lot of material from the inside, from the internal of the cell, and this explains why in proteomics analysis of the coronome we found so many proteins from inside of the cell as well. Finally, we were interested in how the material transferred from one cell type to another cell type. Okay, it's logical that macrophages attack the epithelium independently who is contaminated, whether macrophages or epithelial cells. It's also logical that when macrophages uptake material, they gradually slow down, and then finally, after a few days, they actually completely disintegrate. They have holes inside, so macrophages really die. It's interesting that they sense these composites, bio-nano composites on the surface of epithelial cell. They can uptake parts of these composites and they degrade them, because we can see from a lifetime microscopy that the packing of the material after they eat those structures is different than it was in the epithelial cells. For example, here on this image, this is the structure still on the epithelial cell. The lifetime is longer, so it means that the probes are more apart, while after fresh macrophages uptake those structures, the lifetime decreases, meaning that the probes are much closer to each other. So it means that macrophages degrade organic matter, but then are not able to degrade anything else, so they die. And most surprisingly is that even after the macrophages die, the material is uptaken by epithelial cells again. So we have closed all the arrows in the cycle. So we know that nanomaterial actually cycle between the cells all over the time. So this is now entire mechanism of cycling. So you have an uptake in both sides, in both cell types. You have damage cytoskeleton inside epithelium. At least we have triggered lipid production, and then macrophage attack those structures and they die and they excrete material, and this material flows around, and we somehow symbolically denote this cycle as this image on the bottom here. So we have an uptake arrow, we have a transport between epithelium, from epithelial cells to macrophages, and then we have excretion of the material again. Plus, we could see that there is some signaling from epithelial and from macrophages on the moment when the material is cycling, and this signaling is responsible then for calling for recruiting new phagocytes in the system in vivo. So then what we did, we take 3 time points. So the rate of passivation in the epithelial cell, the rate of dying macrophages, so we call this toxicity rate, and the signaling rate, which can be measured either from in vitro or in vivo. And we use those rates and create the simplest possible model to predict in vivo time courses of the passivation of nanomaterial. This is the red course for the signaling, for the recruitment of new macrophages, and for the viable macrophage. And then from the state of these time courses at certain time points, let's say here, we can now predict where individual nanomaterial lies in this phase space on the left. So this phase space is now include 3 rates: passivation rate, toxicity rate, and signaling. And you can see here that the more dark is strong chronic in vivo inflammation, the less dark is mild chronic, and what lies outside this valley, it's actually only acute or just cell die immediately. So with this, we call this nanomaterial sorter, and at least for metal oxide, we can now predict the in vivo chronic inflammation or either acute inflammation for certain type of nanomaterials. So take-home message is very much in line with the SmartNanoTox approach. So we can make a prediction using in silico modeling and in vitro probing through the new mechanism of action. And that's what we did, where we discover actually a cycling and then passivation or quarantining of nanomaterial to predict a type of inflammation for at least one family of nanomaterial. So I would like to thank all my colleagues in the group, and then all groups in the SmartNanoTox, and especially Tobias from Munich, and Ula from Copenhagen, and Vladimir and David from Dublin. Without them, we would never come so far. So thanks a lot.